everyone, and welcome back to Neuropsych. So we have covered a lot of biology of the neurons. So we talked about the anatomy of the soma, cell membranes, protein synthesis, protein packaging and shipment, transport, and then we covered some little details about the membrane. So how we use proteins in the membrane as channels, gates, and pumps for molecules and ions to go from one side to the other side of our membrane. And then we also talked about how the movement of ions create electrical charges. So if this was confusing to you, please go back to the previous video and rewatch it. You will definitely need to understand that video before we dive into this a little bit deeper. Today, we will talk about the membrane resting potential. And then we will talk about the concentration gradient and voltage gradient within our membrane, and also graded potentials. So here you can see an example of an axon with all these little ions in and outside of the cell. And here we have two examples of graded potentials. Okay, so when a neuron is at rest, there is an unequal distribution of little ions inside and outside of the cell. So at rest, the neuron is more negatively charged inside of the cell compared to outside of the cell. So specifically, the cell is about 70 millivolts more negative on the inside compared to on the outside. So this is what we call a resting membrane potential. So it can vary from about -40 to -90, but we'll just keep it at -70 for the duration of this course. But do remember that it does change. Okay, so there are 4 kinds of charged particles that interact to produce this resting potential. So you have sodium ions, which you can see right here, the chloride ions, the potassium ions, and protein N ions. So these are little proteins with a negative charge. So at rest, the large protein N ions and potassium ions are in larger quantities on the inside of the cell. So you can see that right here. So whenever the cell is just chilling, it's resting, anions and potassium has a larger concentration on the inside. And then sodium and chloride are mainly on the outside of the cell at rest. So there's a higher concentration of sodium and chloride on the outside of the cell during rest. So although the outside area of the cell is technically positive, we normally just follow the convention for assigning it a 0 charge when we talk about it, or when we just discuss it for comparison reasons. So whenever we talk about a membrane, so here you have the voltage on the y-axis and time on the x-axis. Whenever we talk about a membrane just resting, we say it's at -70, so it's just stays here. It's just at rest. Okay, and remember that these anions and potassium have a higher concentration on the inside of the cell, and sodium and chloride have a higher concentration on the outside of the cell when the cell is in rest. Okay, little recap. Protein N ions inside of the cell, potassium ions mostly inside of the cell, and potassium ions actually move out of the cell when a number of them in the cell is higher than the outside of the cell. So it will move down the concentration gradient. And then the other two, sodium is mostly outside of the cell whenever the cell is at rest. And regarding sodium, we talked about pumps before. So the sodium-potassium pump is really important for this. The sodium-potassium pump is a protein molecule that will shunt sodium out of the cell and potassium into the cell. So it works continuously exchanging 3 sodium for 2 potassium ions. So this pump needs a lot of energy, what we call ATP. And lastly, the chloride ions have a higher concentration outside of the cell. So just a little recap. Okay, so channels, gates, and pumps, all three are very important for maintaining this resting potential. So protein and ions remain inside of the cell as there's no channel large enough to transfer these. Potassium is technically free to enter and leave the cell whenever it wants because these are very small, so it doesn't really matter how much goes in or out. And then we have sodium channels that are normally blocked, so unless an action potential happens, which we'll talk about later. But normally sodium channels are closed, so they don't let sodium through. And then lastly, we have the sodium-potassium pump, which pumps 3 sodium ions inside of the cell, exchanging for 2 potassium ions out of the cell. So keep these 3 specifically in mind. Okay, so now we know a lot about the resting potential. So as we go a little bit further in knowledge to our action potential, we have to talk about graded potentials first. So graded potentials are increases or little decreases in the membrane voltage, and they are specifically very localized and restricted to where they are produced. So a decrease in voltage of the membrane is called a hyperpolarization, as you can see right here. And an increase of the voltage in the membrane is called a depolarization, so right here. So during a hyperpolarization, the cell on the inside gets a little bit more negative compared to the outside. And in the depolarization, the cell on the inside gets a little less negative compared to the outside. So it's not positive, but it's becoming a little less negative. It's going from negative 70 to, let's say, negative 63 or something. So it becomes a little bit less negative. So whenever we talk about graded potentials, remember these two. Hyperpolarization, it's a dip down, so it becomes more negative. Depolarization is a little bump, so it becomes less negative or a tiny bit more positive. Okay, so an example of an hyperpolarization could be when there is an efflux of potassium. So technically potassium will go outside of the cell. Since potassium is positive and is leaving the cell, this will make the cell more negative. Another example could be when negative chloride ions go inside of the cell. So either when negative goes in or positive goes out, the inside of the cell will be a little bit more negative. An example of a depolarization could be when positive sodium ions go into the cell through sodium channels. So when positive goes into the cell, it will increase the voltage of the inside of the cell and make the cell a little bit more positive on the inside compared to the outside. Okay, so this was the end of this video. Try to rewatch it one more time, and then in the next video we are going to dive into the action potential.